So I just want to thank the organizers for the opportunity to tell you about some of the latest work in my lab, as well as kind of some bigger picture views of where I see single cell genomics being applied clinically. So just a quick disclosure, I am going to be talking about a new whole genome amplification method developed in my lab that's being commercialized by BioScribe, where I'm a co-founder, equity owner, and board member. So just to start out kind of broadly, I think everyone would agree that genomics has really transformed our understanding of cancer and has really transformed a lot of areas of clinical practice within oncology. And many studies similar to this large target study that I was involved with have sequenced bulk tumors, and that has revealed a number of new mutations associated with different diseases, and really, provided catalogs of details of cancer formation that we knew very little about. However, I think what also quickly became apparent is that tumor heterogeneity is a huge problem. And when we have one of our cells expand into billions or trillions of cells, that creates massive genetic diversity and creates a huge problem, both for kind of profiling the genomics of the tumor, as well as also identifying drug treatment strategies. And so even in bulk sequencing of different locations in the same tumor, in this now kind of classic paper a few years ago, the authors found that there were actually distinct mutations in different locations in the tumor when they did bulk exome sequencing, although they did find that SETD2 mutations, different SETD2 mutations were common in different places in the tumor. But it really kind of shed a light on this concept that tumor heterogeneity is a really big problem. And that has also played out clinically. So again, we're seeing a lot of studies like this, and I've had this experience clinically as well, that when you do broad-based genomic testing and you compare that to routine genetic testing, there are a small subset of patients that benefit from that kind of additional information that you get. But that really isn't as big of a magnitude, I think, that as all of us expected that to be. And so, a large part of that is that we know that the tumors evolve, and there's genetic heterogeneity that's selected for as patients go through treatment. And so kind of the logical next step would be to start to interrogate these tumors within single cells within the tissues to really get a sense of that genetic diversity, but also to identify and catalog variants associated with specific phenotypes that are clinically important, such as drug-resistant tumors. And so I'm not going to talk about single-cell RNA-seq, but this is an example of a project from my lab with Lauren and Yakun. But many labs around the world, with the kind of advances in single-cell RNA sequencing technologies, that begin to kind of dissect the ecosystem and look at the roles of immune cells and other non-malignant cells, as well as differences within the malignant cells within a population based on gene expression. However, I would argue when you start to think about things clinically, almost all the companion diagnostics that are approved right now are either based on mutations or a surrogate of a mutation. So for example, ALK overexpression can be a surrogate of an ALK fusion. And so I would argue that I think this kind of paradigm is going to hold true in at the single cell level, where single cell RNA-seq is a really useful tool for really understanding the biology, and maybe in some cases for things like markers of immune activation and things like that could also be used clinically. But within the tumor cells, I would argue that it will still be DNA that's going to be the most important. And so from a diagnostic and clinical targeting standpoint. And so that's really what I've focused on in my lab. And so why is that? So to step back a little bit, when we develop, we go from a single cell to trillions of cells. And because we acquire a coding mutation every 3 cell divisions, roughly, or a mutation roughly every 3 cell divisions, we acquire trillions of mutations just during the process of development. And in my lab and others have been looking at cardiac disease, brain diseases, vascular malformations in this cases, pulmonary malformations. And we're really starting to learn the roles of these somatic mutations in many different diseases. And so this is just a group that I've collaborated with recently and actually over the past few years But I think this is an area where we're really starting to understand that even in non-malignant diseases, these somatic mutations are important. But that's even more amplified within tumor cells. So again, our cells expand from one cell to trillions of cells, but then our bodies are replacing billions of cells every day. And so we're continuing to acquire copying errors as we age. And even more so, that's even more dramatic in tumors, where again, you're going from one cell to billions or trillions of cells. And so, you know, just based on the number of cell divisions and the background copying error rate, we're continuing to acquire, you know, many mutations within these tumors that we don't see traditionally with bulk sequencing. And so people within the field of single cell DNA sequencing have generally kind of taken one of two approaches. And so kind of on one extreme, you can look at many cells, but a large fraction of the genome. On the other extreme, you can look at a small number of cells, but a lot of the genome. And so the main limiting factor for most labs for this has been cost. And so if you look at a lot of cells and a lot of the genome, it gets very expensive. And so depending on the question you're asking, the labs tend to either look at many cells and focus on, you know, very specific areas, or less cells and lots of the genome. I would argue now that really we're in a phase of discovery. And so looking at more of the genome and discovering new genes associated with specific conditions is really where the field is right now. And then later, you could imagine looking at many cells and looking at the areas that, you know, we've discovered are really important. For example, resistance to treatment in a specific malignancy. And so I would also break down these areas into kind of technological innovation. And so for single cell genomics, there are basically two areas of technological innovation. One is in isolating the cells, and the other is in amplifying whatever nucleic acid you want to look at. And these are the fluidigm chips here. This is an example of microwells, of droplets. And so especially in the single cell RNA-seq world, we've seen that's really enabled us to increase the number of cells that we can look at in a much easier and much more efficient manner. And within this, I would also include now 10x and Mission Bio and some other companies that are out there. On the other side is wanting to look at more of the genome and look at more of or transcriptome in this case. Having really access to more of the genome, again, could enable many of these discoveries of genes that are associated with certain phenotypes within a cellular population. And so that's an area that my lab has been working on over the last few years. And really the bottleneck to kind of the field of single cell DNA sequencing has been the quality of what you get when you do whole genome amplification. And so starting with a single cell, which you need to amplify thousands of times to get enough material enough to sequence, you need to undergo whole genome amplification. And really for a long time, the gold standard has been this method called multiple displacement amplification. But as those who have used it know, it suffers from these issues. This is walking across, in this case, the E. coli genome. But you find this jackpotting effect where wherever you primed your reaction first, you get overrepresentation, and then you get really underrepresentation of many other areas. And so, again, this poor genome amplification uniformity or coverage leads to loss of data during the amplification. There's also issues with reproducibility. All these contribute to cost. And then MDA hasn't really been able to be put into droplets or microfluidics to look at lots of cells. And a lot of it has to do with the barcoding. And so there are a number of challenges around that. There are products out there that do it, but they haven't been able to recover a lot of the genome when they barcode. And so in my lab, we set out to address this problem. And again, going a little bit more details of multiple displacement amplification, you get random priming, and then you get this special polymerase, Phi29 polymerase, that extends, and it has a strong long processivity, so it can go on for tens of kb. And then it also has another feature, which it has strong strand displacement activity, and it can displace the strand in front of it. And that leads to these branching structures. But as I showed you, that leads to overrepresentation of regions that were primed early and underrepresentation of neighboring regions. And so we came up with this new method we call primary template directed amplification, which again is like MDA and takes advantage of the processivity, the strand displacement, the low error rate of the enzyme. But instead of letting it extend for tens of kb, we add an irreversible terminator to the reaction. And that reduces the amplicon size. And then those amplicons get displaced and they go limited subsequent amplification because they're shorter. They can be double-stranded more quickly. And once they're double-stranded, the reaction isn't heated up. So the primers then need to go back to the primary template to continue amplification. And so in theory, this would result in smaller amplicons that cover the genome in a much more even manner. And I will show you, again, just a few slides on our new method that we think is going to be really enabling in the discovery space of cancer at the single cell level. And so you can see with PTA, our method, we can cover, we have, we're not producing much junk DNA. So, 97% of our reads map to the human genome. You can see that similar to MDA and some other in bulk samples even. Some of the methods that use a lot of PCR have much lower mapping rates. But the area when I got really excited was when we look at increasing number of reads and we look at the fraction of the genome covered, you can see PTA is similar to the two bulk samples in this case. And so with the same amount of sequencing, we're covering almost the same amount of the genome from a single cell as we would from a bulk sample composed of tens of thousands of cells. And there are error bars there that you can't see, but it's actually quite reproducible. And you can see significantly better than the other methods, and the error bars are significantly smaller than the other methods, suggesting the reproducibility is much better. And so this is what that looks like walking across chromosome 1. This is MDA. This is the centromere that doesn't get much coverage from 529 or from this protocol. But again, you can see these real big spikes in coverage and these real losses of coverage. And again, that results in sequencing these areas a lot and these areas not much at all. And you can imagine how that hampers being able to call variants. And so you can see PTA is not perfectly uniform. Again, we lose coverage at the centromere, but we don't have those valleys with the same amount of sequencing and we don't have those big peaks. And so we're much better able to call variants with PTA. And so this is how that looks. And so you can see with increasing number of reads, we use the bulk sample as a reference and say what percentage of the variants can we detect, and it's from a single cell that were present in the bulk sample. And you can see with increasing sequencing, we're above 80% for PTA, and the next best methods are down in the 50s. I will say more recently, we've even been able to get it up to over 90%, and there are further improvements on the protocol, as well as the computational pipelines that are further improving that as we've gone. Another interesting feature of PTA is it allows smaller templates to compete with the larger nuclear chromosomes. So you can see with the same amount of sequencing, we're covering the mitochondrial genome at about 400x, again, compared to MDA, which is a few x. And this turns out to have a number of really interesting applications as well. So now that we have these tools, my lab has really turned back and started to ask these questions around, tumor heterogeneity. How complex is the problem? We really haven't even begun to define the challenges that are there. And again, looking at different locations in the same tumor, you see differences. But then when you get down to the single cell level, you see additional differences. So how many mutations are there in each cancer cell? And really, how much does that vary between cells? And does that vary in different parts of the tumor? We haven't even come up with nomenclature for how we really define a clone. We really have no idea how many drug-resistant clones are present at diagnosis. And I think that's something, that would be important to know clinically. And then we really don't know how chemotherapy contributes to the development of treatment resistance. Or so specifically, is it inducing mutations that are increasing the evolution and kind of promoting more broad spread drug resistance? And so we've begun to look at this in my lab in leukemia cells. And this is a paper published from Stefan Pfister's group looking at pediatric samples where they estimate the number of coding mutations per megabase. And you can see people have kind of understood that pediatric cancers have lower mutations per megabase in general, and that leukemias tend to have less as well. But we repeated this, and we used whole genome sequencing instead of exome. But we also isolated single cells from the same sample and did whole genome sequencing. And what we can see is actually our number of mutations per megabase is similar to what was seen in the bulk leukemia samples. But when we look into each cell, we see about 10-fold more mutations per cell. And so if you kind of multiply this across billions of cells present in a patient's tumor, you can imagine the level of genetic diversity is there. And it's no wonder that many cancers we have a hard time treating. We have to treat with many different drugs. And drug-resistant clones grow out, as the norm in many cancers, especially in adult cancers where there are higher background rates, where I would suspect the per cell rates are even higher. And so we've tried to look deeper into this heterogeneity in my lab. So Yakun's been working, taking diagnostic samples, taking samples after 4 weeks of treatment, and then kind of simply asking which clones were selected for, and then doing single cell exome and Cnb calling, as well as surface marker profiling. And the big idea is if we do enough samples, we'll be able to create a catalog of connecting genotype with phenotype. So if we see a clone, for example, or these two clones in a patient sample, and we know from previous experience that clones with these mutations are sensitive to these drugs, but resistant to these drugs, we can then start to devise a rational approach to combining therapy for each of these patients. So in this case, they appear to both be sensitive to this drug and this drug. So it might make sense to target these two clones with these specific drugs. And so to just wrap up with an example of that from my lab. So this is a diagnostic leukemia sample. These are blasts. So you can see 50% of the blasts didn't have a JAK2 mutation, but the other half had one of these two known activating JAK2 mutations. When we sequenced the bulk sample after 4 weeks of treatment, the patient had about 1% blasts left, but we didn't detect any of the JAK2 mutations based on bulk sequencing. When we looked at CND calling, we could see this loss on chromosome 4 that was present in all the cells. And then we see this kind of subclonal loss on chromosome 6. And then again, we didn't see any CND changes within the bulk remission sample. And so we wanted to dig into these persistent cancer cells that are this little sliver here. So we sorted out single cells, again, did index sorting, used our genome amplification, made libraries and sequenced. And what we found was within the MRD cells, actually, most of them had one of the two JAK2 mutations. So again, this was bulk and diagnosis. This is day 29, just the MRD cells. And you can see we could detect one of the two JAK2 mutations in most of the cells. We could also correlate that with surface marker expression, and this is important for many immunotherapies where loss of expression of an immunotherapy target can be problematic. But we did see some evolution of the surface marker profile. Here you can see on day 29, many of the cells had much higher CD34 expression. There was some CD20 expression we didn't see at diagnosis. And so it suggests it was selected for the small population that was CD34 positive. We can then correlate marker expression and genotype. And in this case, we don't see any correlation. We see that both JAK2 variants can have high or low CD20. Most are CD34 high. But then what got really interesting, and I think really kind of brings home some of the points I'm trying to make, are that when we looked into each of these populations, so this JAK2 mutant population, this JAK2 mutant population, they had some SNVs at diagnosis, but then we saw some other SNVs that were acquired or selected for during that 4 weeks of treatment, some of which have potential, make sense in terms of being mediating drug resistance. So PELP2 is a steroid coactivator. And so this could be mediating resistant to steroids, which the patient received for 4 weeks prior to this. But in addition, we also see Cnb evolution. And so again, in all the cells, we see this loss of chromosome 4. We see also this loss here, a group of cells with this loss on chromosome 6. We also see some cells that don't have any additional CNB, and then we see 10% that have other changes that vary across the board. But interestingly, within this population, there's this large clone that has about 40% of this population that has this deletion on chromosome 11, which was not detected at all at diagnosis. So it was highly selected for. Again, within this clone, we have some cells that don't have CNB. And then again, we have a range of other CNB that we see. So I think this just illustrates that, you know, there's a diversity of the genetic diversity is massive that's there when patients are diagnosed, and we're really at the beginning of of understanding that and being able to catalog that and correlate that with treatment outcomes and potentially help, you know, guide better treatment strategies. And then finally, people are starting to look at genotype phenotype interaction. So I showed you just surface marker expression and mutation, but there are protocols coming for doing whole genome, whole transcriptome, whole genome, whole methylome, potentially all three. And we can really begin to answer these questions. How do genetic changes interact with the state of the cell to affect or result in the phenotype of that particular population? So in conclusion, I would say that single-cell genomics is really transforming our understanding of cancer biology. And I really do believe in cancer that's really heterogeneous. Most sequencing in the near future is going to be single-cell based. Tumors are much more genetically heterogeneous than we appreciate, but we still need a lot of work needs to be done to define that magnitude and other features. And again, I do believe that single-cell DNA sequencing is going to be very useful clinically, and that correlating a specific genotype with a phenotype, such as drug resistance, is going to lead to new biomarkers, new drug targets, really a much higher resolution, more personalized approach to cancer therapy. And so with that, I'd like to especially thank my lab previously at St. Jude and then more recently at Stanford, especially Veronica, who did most of the PTA work. But everyone in the lab has contributed a great deal. Thank my funders, and also open to accepting postdoc applications, and look forward to your questions.